Hello, everyone. Welcome to Life Science Across the Globe. My name is Janine Stevens, and I'm one of the Janelia organizers of this series. We are extremely excited for today's event entitled Learning from the Planet to Heal the Planet, Marine Ecosystems, hosted by EMBL. We'll have 3 short talks followed by a moderated discussion and audience Q&A. You're welcome to write your questions in the Q&A box at any time. And then we'll end with a Meet the Panelists session. We invite students, postdocs, and anyone in a training position to stay on the call and ask questions to our panelists and hear from them on career development related topics. I encourage all of you to also please complete the very brief survey that we'll link in the chat box because your feedback is very important and very much appreciated. And with that, I will now turn it over to Dr. Cornelius Gross, Group Leader and Interim Head at EMBL Rome. Cornelius.
Hello, everyone. Welcome to this edition of Life Science Across the Globe that's sponsored by EMBL. My name is Cornelius Gross. I'm the director of EMBL's site in Italy, and it's a special honor for us to be hosting this edition. Maybe just a few words about EMBL as a hosting institution. EMBL is an international organization. We're a little bit different than many research organizations. We have a mission to carry out fundamental research. So we have about 100 labs at EMBL across 6 different sites in 5 different countries in Europe. But then we also do lots of service work. So support for researchers within EMBL, but also outside all over the world to provide them with things like imaging expertise, bioinformatics expertise and database management. We run beamlines, for example. So we have this dual role of service and research, and we combine those 2 together to make sure that they each inform each other. So today's topic is close to our hearts. It's going to be about marine ecosystems. As you just heard, we're going to have 3 really outstanding speakers, and I'll hand over in a few moments to Justin Crocker, who will introduce them to you. But marine ecosystem research, as I just said, has been very close to EMBL's heart because EMBL has had a long tradition of supporting research in this area from national institutions, again, in the areas primarily of imaging, of bioinformatics, and of molecular biology in general. So we're really looking forward to this. I hope you enjoy it. I hope you will stay also for the various interactive sessions and contribute. And let me hand it over now to Justin Crocker. Justin Crocker is a group leader at EMBL. He runs a laboratory in Germany, in Heidelberg, and he will tell you more about today's speaker.
All right, thank you very much, Cornelius. Yes, I'm very much looking forward to the discussions throughout the talks today. And I just want to point out again, as you have any questions, feel free to post them throughout the discussions, just because it keeps things going and we'll address them towards the end. So without wasting any time, because I'm really looking forward to hearing from some of these, the luminaries in the field who will be discussing some of these, I think very important topics. So I'd like to actually welcome the first person who will be talking, who's Minhan Dai. He's a professor of marine biogeochemistry from Xiamen University in China.
Thank you, Justin, and thank you, the seminar organizer, EMBL. And today I'm going to share with you some of these on the ocean carbon thing. The title of my talk is Potential Synergy Between Mitigation and Adaptation for Ocean Carbon Think. Hope it's doing okay. A big context of my talk is probably you're well aware of this, the injection of anthropogenicity into the atmosphere and its subsequent distribution across the ocean, land, and atmosphere components of Earth system that led to unprecedented perturbation of the global carbon cycle has been ongoing since the Industrial Revolution. So the second important context is many countries has adopted implementing the Paris Agreement. So in reduction of carbon neutrality related emission, that will introduce another set of perturbation to the Earth system on an even short time scale. So today I'm trying to walk you through a little bit the ocean carbon thing, and I will show you the temporal evolution of the ocean carbon and the marine CDR, that's marine carbon dioxide removal. That's a very hot topic in the field. And then try to bring together the synergy between adaptation and the mitigation. For those who are not familiar with the ocean, those are the 7 literacy of the ocean science. The Earth has a big ocean with many features. The ocean and the life in the ocean shape the feature of the Earth. The ocean supports the great diversity of life and ecosystem. The ocean makes Earth habitable, produce 50% oxygen flux, dominate the water cycle, and provide 80% habitable space. The ocean is, of course, a major influence of weather and climate by uptaking close to 90% anthropogenic heat and 30% anthropogenic CO2. The ocean is largely unexplored. After all, the human and the ocean are inextricably linked together. So I would argue this a powerful word, phrase: the Earth needs the ocean as life needs the water. We're all familiar with the anthropogenic CO2 emitted from fossil fuel burning and the land use change. That's the left column. And the thing of this anthropogenic CO2, including the atmosphere, that's causing the greenhouse effect. And then the rest goes into the terrestrial ecosystem and the marine ecosystem. And if you look at, stay back to this pre-industrial era, the atmosphere CO2 start from 280, and then you add up different sources by fossil fuel burning, coal, oil, you name it. And if without ocean and land, think atmosphere CO2 now would be close to 600 ppm. That's already 50% higher than the actual value we are measured. So the land and ocean ecosystem thing is of paramount importance. Just these slides give you a broad view of this major carbon reservoir on the Earth's surface and the flux of the global carbon cycle. There are 3 key messages I want to convey from this diagram, schematic diagram. When the ocean contains 90% of the carbon in these reservoirs, and the ocean was the net source, emit CO2 to the atmosphere at the range of about 0.6 petagram carbon in the pre-industrial era. Now, the ocean is a very significant net carbon sink. If you measure the CO2 flux, it's about close to 2 petagram carbon. If you add in the pre-industrial source, then now the contemporary ocean is a sink of 2.5 petagram carbon per year as an anthropogenic carbon sink. And that gives you another view of this ocean flow in the carbon cycle. The left panel, actually, again, during the last 50 years, the ocean absorbed about 25% of the anthropogenic CO2. And the right column tells you if you only count the fossil fuel burning emission, because on the land use emission and the land ecosystem uptake is practically offset each other. So we argue that ocean is the only sustained thing of anthropogenic CO2 for the last 150 years. I don't want to give you too much details of the carbon cycle, but essentially this gives you how the ocean functions as the moderator of atmospheric CO2. Start with the gas exchange between air and the ocean, and then followed by a suite of the chemical equilibrium reactions of the carbonate system. And then part of the carbonate consumed by photosynthesis, that drives the biological pump. That's the left part. And then the solubility is driven by the temperature, salinity, and the pressure. This drives the physical pump. So if you look at the flux equation, it's a function of the gas exchange coefficient, solubility, and the PCO2 difference between air and the ocean. So if you look at the solubility, it determines of the solubility pump. That means the high temperature dissolves less CO2, and the photosynthesis with the uptake, that drives the PCO2. So both the solubility pump and the biological pump drives the CO2 flux into the air, into the ocean. Just give you the idea how the ocean breathes the CO2. The up panel is the coastal ocean, and the left lower panel is the global open ocean. The cool color that signify the carbon sink, and the warm color is the carbon CO2 emits into the atmosphere. So there's a strong latitude pattern, overall strong, very strong sink in the high latitude, except in Southern Ocean. And in the lower latitudes, you see the CO2 as a CO2 source to the atmosphere, or being equilibrium between ocean and atmosphere. With those rising PCO2 and the climate change, those have fundamentally changed the marine ecosystem with current shift in temperature, you name it, warming, circulation patterns, stratification, oceanification, and the deoxygenation. Those are the symptoms of this of the atmosphere CO2 rising, and so the ocean is both as an actor of against the climate change, also a victim of the climate change. So I just adopted this IOC, the Integral Ocean Carbon Research Report, just released a few months ago. Those listed the overarching question in ocean carbon research. The first one will be will the ocean uptake of anthropogenic CO2 continue as a primary, as a abiotic process? What is lower biology is very much relevant to this seminar. And what's the exchange of carbon between land and ocean continuing, and how they evolved the ocean over time? And the most important, how the human are altering the ocean carbon cycle, resulting feedback. So including, I will touch a point on this possible purpose-free carbon dioxide removal from the atmosphere. With that, I'm trying to go into show the temporal evolution of the carbon and the CO2 in primary. This gives you an idea of the two time series station. One is in the Pacific, and one is Atlantic in the subtropical region. You can see clearly the gray is the line is the atmosphere CO2 availability, and you see these both at the Pacific, Atlantic. So essentially, the ocean PCO2 follows very closely with ocean PCO2. So that's one of the reasons the ocean has been uptaking CO2 as a function of solubility. It's an abiotic process, it's a predominant process. If you move into the coastal ocean, this is the Baltic Sea, Mid-Atlantic bites, and the South China Sea. You see much higher amplitude availability of PCO2 in the ocean, in the surface ocean. Extremely variability in the Baltic Sea because it's very close with strong biological activity. This is the seasonal pattern. And if you move Atlantic, in the Atlantic, Mid-Atlantic bites, it's much less variable. And if you go into the South China Sea, big marginal sea, essentially similar to the open ocean. So I would like to take you back to another view to look at how the ocean carbon has evolved since from the pre-industrial era to the contemporary era, and then into the future. This schematic essentially shows the pre-industrial, the natural carbon cycle. So essentially, the land was a small thing, and then the organic part of the organic carbon discharged from the river into the coastal ocean and the open ocean, about at around the 0.6. So it's essentially those organic from the river and from terrestrial system were expressed and essentially degassed. So during the pre-industrial era, the ocean is a source to the atmosphere CO2. So the red arrow, this shows the present time, and you see anthropogenically altered CO2 fluxes. Essentially, the ocean becomes a very strong CO2 sink from the atmosphere. In particular, if you look at the coastal ocean, the alteration is, the percentage of the alteration is very big. And so that's one of the vulnerability of the coastal ocean in response to the anthropogenic CO2. If you move into in the future scenario, and you immediately notice a lot of question marks. but essentially you continue to see the alteration of the anthropogenic CO2 into the coastal ocean. But if you look at the flux number, it's much less from the now. Looking into the future, it's quite difficult to predict. But essentially, the Earth system model suggests significant weakening, even potential reversal of ocean lengthening under future low emission scenario, essentially driven by the PCO2 in the air, because the essentially driver into the land and the ocean is the delta PCO2. And the prediction, why it's so difficult for predicting the future trend, because one of the reason is the land, ocean, and atmosphere coupled system. So on the left column, you see all the different drivers from the land, river dissolved inorganic carbon, nutrient driven by the fertilizer usage from the land. On the right column, essentially the ocean boundary will change with the exchange with the coastal ocean and open ocean. So in sum, it's very difficult to predict future carbon thing. So I'm trying to summarize the past ocean evolution and the possible future scenario. Again, the human being has been doing the experiment by injecting the fossil fuel CO2 into the air, and the ocean has been a sustainable thing of anthropogenic CO2 over the past 150 years. And adaptation of the implementation of Paris 6 will introduce another set. And we'll see continued emission growth followed by the transition from the peak around 2030 and the net zero emission around in the middle of the century. Then the air CO2 will peak in the 2 or 3 decades and will start to decline. So the important message embedded these contexts are to maintain atmosphere CO2 and temperature at low level. Not only does anthropogenic CO2 in the atmosphere need to be removed, but also anthropogenic CO2 stored in the ocean and the land need to be removed. Otherwise, it will degas back into this atmosphere. So that comes the concept that the marine CDR, marine carbon dioxide removal, essentially through 3 ways: chemical conversion and biological conversion and the physical transport. And if you go, I'll give you just 2 examples. One is the chemical conversion by so-called artificial ocean alkalinization. Essentially, if you add the alkalinity into the ocean by using, for example, olive mineral, the dissolution will add alkalinity into the seawater, and that will increase the capacity, buffer capacity of the seawater and enhance the CO2 uptake. Another example I will show you is one of the projects funded by National Science Foundation of China. So essentially, I have been looking at the subtropical dry, I call the ocean desert, extremely eutrophic system. Those systems extremely poor in nutrient, but potentially nitrogen fixation can be shipped in, and it's limited by iron fertilization. Iron. So if you use adding iron, will potentially stimulate the nitrogen fixation and enhance the carbon sequestration in this. So the ratio of those iron, nitrogen, and the carbon extremely important. So this marine CDR against the marine carbon dioxide removal, the core benefit is of course the mitigation of the atmosphere CO2, but also keep the ocean in synergy with atmosphere in terms of CO2 level. Also, CDR in marine system doesn't consume the land. So the land use, it does not change. There is also co-benefit, for example, de-acidification, if you add alkalinity mineral into the ocean system. Of course, there is many issues, research needs and the policy needs in this just the emerging field. So finally, I will try to put together the synergy when we adopt the techniques or approach or policy between mitigation and adaptation. we really have to think about the synergy and think about the ecosystem-based and consider. So when we reduce the greenhouse gas emission to minimize ecosystem consequence, ecosystem-based CDR mitigation pollutants, those are the synergy when we think about the mitigation approach. Of course, we have to think about the adaptation as the synergy of the mitigation. So the management, sustainable climate, smart management, for example. Of course, if we look at it, incentivize the ocean carbon ecosystem. I just to give you one very dramatic example, because we all know the mangrove is a very significant, strong carbon sink. But this example gives you another source about the ecosystem approach, because this is a mangrove used to be a muddy wetland located in northwest Taiwan, and they started to plant mangrove in the late 60s, and until 2000, the year of 2000, the mangrove covered a large area. But they find the serious problems because it changed the structure and the function of the habitat for benthic organisms. Most important, the sediment, because Taiwan is located in mountainous river, the sediment discharge is blocked by the mangrove system. So in the year of 2000, they started again to remove all these planted mangrove. So I would highlight this carbon thing is only one of the crucial ecosystem service. We have to think about the integrated ecosystem-based governance. And so this is what's the message I want to show you from this small example. I think I conclude by my talk with, I don't want to read it again because my time is up, but essentially ocean has been very important, but the predicting the future ocean carbon things is a big, huge challenge. With that, I thank you all very much. I hope my time is about right. Thank you.
Great, thank you very much, Minhan. That was a very nice talk. So I think now we're going to move on to our next speaker in the interest of time. So I'd like to introduce Nicole, who's the director of the Department for Marine Microbiology at the Max Planck Institute right here in Bremen, Germany. So why don't you take it away, Nicole?
Thank you. I'll share my screen. I think I'm fine now, right?
Yep. Okay.
The only thing I don't see is the timer, but I'll just use my iPhone. So I'm fine. So most model organisms in the life sciences are not marine. And here's a recent, actually recent 2019 tweet from EMBL. I hope that whoever tweeted this at EMBL will forgive me. They got actually quite some flack immediately with people saying, well, what about plants? And what about fish, zebrafish mainly, which are freshwater? And what about frogs? I don't see a single marine organism here. And there was in 2015, admittedly, but until very recently, an actually beautiful volume of articles about model organisms, and not a single one of these is marine. Well, these are really, it's a shame because the highest diversity of life, of course, is in the oceans. There are many, many, many more phyla in the oceans. This is from a recent summary of all the microorganisms, of all the plants and animals that occur in the oceans. And so, of course, we very much restrict our understanding of biology and the life sciences by focusing so strongly, at least in the past, on the small number of genetically tractable, mainly terrestrial organisms. But these are wonderful times to be a marine biologist in the life sciences. That recognition has reached many institutes. These are just some examples of a program called the Aquatic Symbiosis Genomics Project that Wacom Sanger, together with the Moore Foundation, has put together. And you'll be hearing in the talk after mine the emphasis that EMBL is putting on marine research and planetary biology, and also working together with the Tara Ocean Foundation to look at our oceans. So I'm going to tell you about the research that we do in my lab. It's on chemosynthetic symbioses, and these were discovered only 45 years ago in the deep sea. And I say only 45 years ago because that's still in the lifetime of the scientists that were around when they discovered it. And it's also the last major ecosystem on Earth that has been discovered. I argue that we will not discover any large ecosystems like the ones in the deep sea that were discovered thanks actually to technology submersibles. These are gigantic ecosystems. The researchers were extremely surprised to see such large animal ecosystems in the deep sea because they thought these were always dependent on light. The biomass is tremendous, as large as tropical rainforests or coral reefs, and the majority of this biomass is based on microbe-animal symbioses. And the reason for that is because the microbes can use the energy sources at the vents that the animals are normally, that animals can't use. And I'll explain that in a second. That's chemosynthesis. What I'd like to emphasize is we discovered these amazing symbioses in the deep sea at 3,000 meters water depth off the Galapagos Islands, and then soon discovered that they also occur at cold seeps, whale falls. But what is remarkable in retrospect is that they're actually in very shallow coastal water sediments, and zoologists had often wondered why animals didn't have a mouth or gut and had some very odd structures in their bodies. These were symbiotic bacteria. They couldn't interpret them properly until the deep sea symbioses were discovered and people went back and looked at them in more detail. So how do these symbioses work? In photosynthesis, it's the energy from the sun that is used by chloroplasts, which are essentially also symbionts or derived from cyanobacteria. And they use that light energy to fix CO2 into biomass, and that's transferred to the plants. What you have in chemosynthesis is that the chemical energy that, for example, seeps out of the hydrothermal vents, reduced gases like hydrogen sulfide and methane, they are used by the symbiotic bacteria to fix CO2, and that is then transferred to the animals. That's why we call it dark energy, because it's independent of photosynthesis. What you're seeing here is a tree that we put together of all the different bacterial lineages that have formed symbioses with animal hosts. And what we now know is that these types of symbioses are not rare. They have evolved independently of each other numerous times in convergent evolution. At least 15 bacterial groups have been able to come together with hosts, and at least 2 protists and 9 animal groups have established these symbioses. And what that tells us is there's a strong selective advantage for both partners in coming together in these associations. These are some of the hosts that we study, and these the questions that we ask are ones, we're organismal biologists, so we try to understand the entire organism in its setting. First, how do the symbiotic partners interact? How do they interact with the environment, and how does the environment affect the symbioses? And what are the evolutionary processes that have shaped these symbioses? And we use a wide range of tools, field work when we go out and collect them, quite a bit of imaging work that I'll be showing in a few slides from now, incubations in the lab and displacement experiments, and then omics, which I'll also present briefly. For the rest of my talk, I'm going to focus in on the deep sea mussels that we work on. They're called Bathymedialis muscles. And so far, nobody has been able to cultivate chemosynthetic symbionts. But what we can do is keep the hosts in aquaria over many years and manipulate them. And I'll first show you the imaging work that we've been doing on these symbioses. And these are the researchers that have been doing it, Nicole, Benedict Gaia, and Max Planck in my lab. So this is a microcomputed tomography image of a deep sea Bathymedialis mussel. And the gills are the symbiotic organ. They carry the symbiotic bacteria, and they're greatly enlarged compared to their shallow water relatives, like the blue mussel, that don't have symbionts. And the reason they're so enlarged is they're packed full, and I'll show you images, they're packed full of bacteria. They have, we estimate about 10 to the 12 bacteria cells in a single mussel of only 8 centimeter size. And they only have 2 bacterial species, one sulfur oxidizer and a methane oxidizer. So they're like a real chemostats that you can look at and study in detail. Let's zoom into what they look like. This is a muscle on a half shell. When you open it up, these 2 dark structures are the greatly hypertrophied gills. And now what we're going to do is look at a cross section through a single filament here of one of the gills. They are composed of thousands and thousands of gills. And we use fluorescence in situ hybridization to look at the symbionts. We use a probe that's specific here to the sulfur oxidizing symbiont. And you can see that it occurs here in yellow in the host cells. And there's a methane oxidizing symbiont here in purple. What you can also see is that immediately adjacent to these symbiont-containing cells, the bacteriocytes, there are also cells that the bacteria don't colonize. And this is very cool. Even within the same symbiotic organism, there's a clear differentiation where the host is saying, I'll let you colonize some cells, but I won't let you in other cells. And these are mainly the cells they use where they're packed full of mitochondria to beat their cilia and to get oxygen. We're going to zoom in now into a single host cell, into a single bacteriocyte using electron microscopy. What you can see here, and I'd like to actually explain this a little bit better, each single filament here is surrounded by seawater. And inside is the host hemolymph tissue. So it's like an epithelial tissue, a single-celled epithelial tissue. And this is the outside here of the cell where the seawater is filtered through by the muscle and the symbionts sit inside the cell. The symbionts here, we're zooming in again. What we have inside here is the hemolymph. Outside here are the epidermal cells, the epithelial cells of the gill tissues. This is the seawater here on both sides of such a filament. And these black blobs are not bad electron microscopy. They're actually gigantic phagolysosomes. And what happens is that the tissues have been modified so that they digest these symbionts. They're eating their symbionts. There are other adaptations. We know that the epithelial cells lose their microvilli. It's called microvilli effacement. And this is a process that is well known from pathogenic bacteria. It's hypothesized that pathogenic bacteria enter host cells and actually cause the loss of these microvilli to close the door behind them. We also see that the gut system becomes greatly simplified compared to the shallow water mussels that have to spend a lot of energy filtering plankton. That's hard to eat. That's like grass. It's a lot of hard work. They've got a looped intestine. And we see that the intestine here has become very, very straight in the mussels because they're getting their nutrition in a very easy manner. So one of the questions in imaging that we've zoomed in is, are these symbionts really intracellular? Because it really matters for the entire understanding of the association if they're intracellular or not. And one of the things we notice in electron microscopy were these openings, very small openings of 50 to 70 nanometers, sometimes 100 nanometers. And we said, well, that's interesting. Are they actually open to the outside environment? Because these are such small openings, you can't really use two-dimensional sectioning to go through them in a 3D manner. And so we were very excited after I gave a talk at EMBL that we had the opportunity, together with Yannick Schwab and Nicole to use their FIBSIM, their focused ion beam scanning electron microscope, to scan through an entire deep sea mussel cell. And for those that are not familiar with this, it's very time consuming. It takes 3 days to go through a single host cell. And it's actually, we're still working on the data. So I think 4 to 8 weeks of analysis time is an underestimate of the wonderful data that we get. So this is what we see. We're now a FIBSIM bacteriocyte, and we're looking at it in 3D. It's now we're going to zoom into the surface of this bacteriocyte. And now in red, you're going to see all the little pores that are there. We estimate that there are thousands and thousands per host cell. The neighboring cell does not have symbionts. It does not have these pores. It has its microvilli, but these pores or openings are specific to the cells that contain the symbionts. And now we're going to go down in a cross section into the host cell. You can see that these are openings. They're clearly opened to the outer seawater, and the bacterial symbionts, false colored here in green, are sitting in a tunnel space just below the outer surface of the host cell. And so you could say, well, who really cares, because, whether they're really intracellular or in this tunnel system, what does that really mean? It means a lot in terms of thinking about host microbe interactions. That we now know that the symbionts are extracellular, it means that they are physically intimate with each other. This means that there's huge opportunities for genetic exchange, just like the human microbiota in the gut, and they have direct access to the seawater circulating in the muscle, so exchange also to any of the components that are in there. There are something like 5,000 openings in the host cell, and what we're very interested in now finding out is what are the cytoskeletal proteins on the host side that can induce this tunneling system. As far as we know, nobody has seen these types of openings before in an animal host. I'm going to move on for the last part of my talk. Let me just check my time because I still can't see the timer, which is fine. I have 5 more minutes to the omics that we do. And this is the team of wonderful researchers that have been working on our omic analyses. And what we are trying to identify is go below the species level with symbionts to the strain level. And I just like to emphasize that's not trivial, particularly if you can't cultivate organisms, as is often done when people are looking at strain diversity. And so we're using metagenome, assembled genomes, and analyses to look at them in more detail. And we're looking at 4 different host species that we've collected on the mid-Atlantic ridge, hydrothermal vents that occur in the middle of the Atlantic. Based on 16S, which is the characteristic gene that is used to look at microorganisms, we know that each mussel species has a single mussel symbiont. But 16S is a really conserved marker, and it's not showing you the type of diversity that really matters in the evolution of animals at a time scale that is relevant. That's why it's so important to also go down to the strain level in microbiology. And what we found with Illumina and PacBio sequencing is that, and by the way, this is for the sulfur oxidizing symbiont, so forget the methane oxidizing symbiont for the rest of the talk. For the sulfur oxidizing symbiont, as many as 16 strains can co-occur in a single host individual. And when we first presented these results, the evolutionary biologists said, well, that really can't be what you're seeing, because our theories predict that in these types of intracellular symbioses, these strains having multiple strains means they would compete with each other for space and resources to the detriment of their host. So how can we explain it in these mussels? We know from the wonderful work that is increasingly being done in high resolution on host microbe associations, including the human gut, what matters is where are the organisms located, host genetics matter, and environment. So as I presented, our imaging analyses indicate, first of all, that they may have access to the environment that may shape strain diversity. It is also possible that there's a spatial separation with each host cell harboring only a single symbiont strain. And we're just in the process of doing PacBio sequencing of single host cells in an attempt to resolve whether that is true or not. What we do know is that host genetics does not play an important role, and I'll just briefly explain to you, there's a hybrid zone where a species from the north and south hybridize, and what we know from their phylogenomic analyses, but also from single nucleotide polymorphism analyses and presence and absence, is that genetics, at least in short-term evolutionary time spans, don't play a role. And so I'll finish up actually with saying what does play a role, and that is the environment. What we know is that there are, this is mapping of Illumina reads to a consensus sequence of the symbiont in a single mussel individual, is that there are regions where not all of the reads map. These are low coverage genes, they're strain specific. And 80% of these genes we were not able to annotate. That's typical for metagenomics, but we lucked out with the 20 that we could. And these are genes that will be critical for the ecology and evolution: virus defense genes, toxin-related genes, and metabolic genes. And I'm emphasizing that these are expressed genes, and many of them, by the way, acquired through horizontal gene transfer. And so these are just some of the genes, and I will finish up with the last 2-3 slides showing why the environment matters with one gene that is used to use hydrogen as an energy source. We were able to show this in 2011, that in addition to sulfide and methane, these muscle symbionts can use hydrogen. And what we see in the strains that in the muscles and the different strains is that hydrothermal vents with very high hydrogen concentrations, this is a geological process that I won't go into now, 100% of the strains have the genes that allow them to use hydrogen as an energy source. At another vent site where the hydrogen concentrations are extremely low, only a small percentage of the strains in each muscle individual have the genes that allow them to use it. And at Lucky Strike, there are intermediate hydrogen concentrations, and there we see that about 50 to 75% have it. So we also imaged this and could show that indeed there's a variability in the strains that have them. And I'll finish up. Let me just make sure, because I still don't see the timing, but I think I'm actually 30 seconds over. So I'll finish up with just summarizing the advantages of strain diversity. The host is able to continuously exchange symbiont strains, both with co-occurring hosts and with free-living bacteria. And it allows the muscles to adapt to the environment within their own lifetime. They don't have to wait for evolution. And I'd like to thank the many people in my lab, in my department, and the sources of funding that have supported this research, and you for your attention.
All right, beautiful talk there, Nicole. I could ask many, many questions right now, but yeah. So we'll save that for the Q&A session after. At this point, then, I'd like to go for our final talk of the discussion today, which is from Guy Cochrane, who's the head of the European Nucleotide Archive, which is at EMBL's EBI, or the European Bioinformatics Institute. So Guy, looks good?
Great. Well, hello, everybody. Thanks very much for the introduction, Justin, and thanks also to all of the organizers for the invitation. It's a great pleasure to be able to be part of this series. So the scientists today looking at marine environments, looking at life in the oceans, has at their disposal a huge array of really powerful technologies to understand, to analyze, and to probe. These technologies are high resolution, they're deep, and they are high throughput. They're very broad. So you could consider sequencing that we just heard about, DNA sequencing, where one can very deeply profile organisms and resolve. On the imaging side, we've seen lots of beautiful images already. We have microscopy, we have all sorts of high throughput and high resolution methods available there as well. Of course, for people studying living things within the oceans, it's important to know the abiotic components of the environments in which they live. And of course, in parallel, outside the biology world, there's a whole host of technologies that have really come of age to provide access to environmental data, oceanographic data, hydrology data, and so on. Think of the advances in autonomous sensing, streams of continuous data. Think of the advances in remote sensing. So for the marine scientists, faced with these very complex, very deep, and very broad, very powerful data sets, there are great opportunities. In order to benefit from any of these data sets, the work cannot be done immediately. No one study that generates data really can any longer expect in itself to fully resolve all of the science that will come from the data set. There's simply a lot more value in there. And so what can happen is over time, whole communities of scientists can work on the different data sets to gradually extract the value and learn science, derive knowledge from those data sets. So what's become extremely important is that one takes an approach to these data that is highly organized and structured and appropriately shares data, preserves data, and makes data available and reusable by whole scientific communities. So what I want to do is focus on the data I'll start first by introducing Tara Oceans, a project that's been mentioned already. This is an initiative in life science investigation of the oceans that I've been fortunate enough to be involved in, to get involved in a few years ago. I'll talk a little bit about the approach that Tara Oceans has, and I'll talk about the data that it produces, and then I'll switch over to talk a bit more generally about the journey that these scientific data take through what is a broad global open data infrastructure for adding value and making these data accessible and usable. And then I will cover such items as data standards and access to the benefits that arise from the data and the science they represent. And then I want to flip back to the Tara initiative at the very end to highlight some of the impacts, some of the outputs that Tara has been able to achieve by taking this structured approach to the data. So this is the Tara, the vessel. She is a vessel that's been in scientific research for several decades. In the last 10 years or so, that's been dedicated to the study of living things. And on the right here, you can see some of the, I move that, there's a thing blocking, there we go. You can see some of the missions, typically voyages that Tara has been on. The current mission is Mission Microbiomes. The ship is somewhere, I think, off South America at the moment. When I give some statistics and some examples later on, I'm going to be talking more about a previous exploration, a 3-year global circumnavigation that you see here, where each of these grey areas is an event. So there's a huge number of sample points along the way. So Tara is ambitious. She concerns herself with primarily with the plankton, the planktonic layer, but at many scales of organization. So there is an interest in understanding the evolutionary relationship between organisms, understanding the biochemistry, the way in which, or the function of individual organisms, understanding how one organism interacts with another, and how you build that up into whole communities, and ultimately scaling all the way up to how communities and whole ecosystems contribute and respond to other planetary systems, how they contribute to their health and changes on planet Earth. So here we're looking at the methodology. So Tara goes on a voyage and stops at various sites and takes samples. So the sampling of the water column is at various levels. So it goes down to almost a kilometer using different types of instrumentation. And concurrent with the sampling, there are always measures of various physical and chemical oceanographic parameters, temperature, pH, and so on. So there's a whole host of environmental parameters that are captured with samples. But the samples are preserved and filtered, size filtered and preserved in different ways for later sequencing analysis and imaging analysis. And on the sequencing side, it's a diversity of different sequencing applications. So here we have the isolation of organisms or the amplification of individual organisms and the derivation of genomes from those organisms. Community DNA studies where you take anonymous samples of large numbers of organisms and you mix their DNA together. You have a mixture of DNA. And you can study that using metabarcoding, which is where you identify, you look for occurrences of certain organisms or abundances of certain organisms based on reference sequences, so certain loci that are characteristic of species or strains. Metagenomics is where you reconstruct whole metagenomes from fragmented reads from these anonymous populations of molecules to reconstruct the individual genomes of what was there. Metatranscriptomics on the right here is where you take RNA, you do RNA sequencing, and you start to look at the activity of the different genes that you find in a particular sample. And then from that, you can infer or begin to infer the biochemistry, the function, the activity of that particular community of organisms that you've been looking at. So in parallel on the sequencing side, there are a whole host of different applications at different scales. We have the very high resolution microscopy, we have flow cytometry, and then slightly lower resolution, but probably higher throughput. We have lots of morphology data available from a host of other methods. So Tara collects many samples, captures environmental data, from those samples, captures sequence data and captures imaging data of all sorts of different types. And just on the bottom right here, we see the kind of organization for the data themselves. There is a sample, there is a registry of samples, there are metadata relating to the sampling process, and then to that we attach the environmental data, the sequence data, the nucleotide data, and the imaging data. And so right from the start, Tara has taken an approach to proactively structure, organize the data, and make the data available as a community resource. So from the start, this has been the data release policy to make data available as soon as possible. So raw data from the images, from the sequencing, and then the early analysis on those data sets. Much of this happens before there's any scientific interpretation, before there's a scientific publication, literature publication. And then there's an ongoing effort to curate the data to make sure that the descriptions of the samples are correct, and to enrich the descriptions of the samples with further information that is external. And so one part of organizing data well is to follow data standards, but the kind of interdisciplinary cross-domain data standards that were required, of course, needed to be developed. So the TARA consortium, amongst other groups working on marine sampling, got together back in 2014-15 to really try to pin down a way of describing and structuring a description of marine data sets for the purposes of the kind of study that TARA and other initiatives were taking on. And this is the M2B3 data standard. It really defines, it doesn't define how you do your science, it defines how you describe your science in a way that allows people to discover, to compare, and to contrast, and so on. And really it's an intersect between details of a sampling process, details of that environmental, that contextual information when a sample was taken, but also information about the sort of large observational biodiversity information that you would want to bring in and intersect as well, and various other things. So incidentally, this standard remains in use by the Tara Oceans efforts as they continue, but it's been adopted by other groups, by many other groups, and it's been informative. So people have certainly been in designing ways of handling their data in more recent studies. It's been informative for that process as well. And so here, just looking at some of the numbers. So this is from back in 2015. So these were the first wave of publications and analyses that came out of the global circumnavigation. So it was a large number of samples overall, 35,000, and thousands of data sets. What was then, this has been very heavily surpassed, but what was then a very large volume of data. And of course, this huge potential to discover many different things. And in the first analysis, looking at the prokaryotic section, there was an identification well, there's a creation of a catalog of some 40 million genes that have been identified. So just to show that this really is science, marine science at scale, and a resource that's made available and can and has been mined long since. So this gives me a chance to switch over. As I switch over to the data part of this, I've become a little bit more generic. This is not just about TARA, it's not just about marine data. But this gives me a chance to introduce my part of EMBL. So this is the European Bioinformatics Institute, and Cornelius has already given the introduction to EMBL, so I'm grateful for that. And he mentioned the service function that we have. And what we're seeing here are many of the data resources that make up the informatics, the bioinformatics data services function that we operate from the European Bioinformatics Institute. And in here we have databases, tools, analysis systems, support, training, and so on. And I'm going to point out in particular ENA, which is the European Nucleotide Archive. This is the sequence database. It's the global, the European part of a global database of record for sequencing data. We have a sample database. We have Magnify. I'll mention this in a minute. This is about metagenomics analysis and many other databases. So typically at EBI, we run the European part of global deposition databases, repositories of data, and we provide layers of analysis and service and value add on top of those to make these data really accessible and usable to people. So putting data in a database is really just the first step. So Tara will put her data, here I'm taking the example of sequence data, into the ENA system, and immediately there will be a propagation, a dissemination of those data. The data gain added reach. And so here we're looking, this is just within the one institute, just within EBI. We're looking at the different data resources that we operate. This is the ENA. And every time you have a band here, this represents a data flow between two different data resources, two different databases, or a data flow or a cross-reference or a link of some sort. And so as soon as the data are put into a database like ENA, there is an immediate and fairly autonomous propagation of those data into different databases. So this is a protein database. This gives us information about a particular protein from the TARA data set. These are protein functional domain databases. They give us information about how proteins are structured that come from that TARA set. This is magnified. This is a set of analyses, visualizations, and tools to interpret metagenomics data, for example, and metabarcoding data and so on. So as soon as the sequence data appear from TARA, they become available through these systems. And it's fairly autonomous because these are established just to do this. So that's within EBI. When we look outside the Institute, of course, there are many, many more layers of databases. The ENA is part of the European e-infrastructure Elixir, which is the coordinating point for bioinformatics data resources in Europe. It is connected through a partnership with NCBI in the US and DDBJ. And there are many other generalist and different databases that will pick up the data, do some curation, add some value, and make the data available in different ways to new people. And of course, some specialist databases here, we're looking at the Marine Metagenomics portal from Norway. This is general for marine metagenomics data. Then we have specifically for TARA, a project from France that builds atlases, gene atlases, barcode atlases. These are just representations. There are many of these. It's difficult to estimate, but there are possibly several thousand databases of different types that pick up these data that come from projects like TARA and make them more usable and more available and more accessible to people. So this is a project on the European side. So Europeans will know that the European Open Science Cloud is the major vehicle through which the European Union will provide scientific data infrastructure for its scientific programs. And here we are building in one project the blue cloud. So this is the marine and the other aquatic informatics services to deal with marine science and aquatic science. And so this is bringing in the sequences, it's bringing in images, but it's bringing in all sorts of other marine relevant data types as well, and providing such services as data discovery and access, virtual research environments, and catalogs. And this is only really possible because we've had a structured approach to data. And the TARA data will, of course, be available through here. And so on the right, we're looking at a demonstrator of some of these services that are being built. This is called the Plankton Genomics Demonstrator. And what we're doing here is, to start with, we're looking for environmental correlates. So we're looking at species distributions, and we're looking at the environmental conditions with which those distributions correlate. And then we're taking data from elsewhere in a second step and extrapolating. So sampling, of course, is sparse across the world's oceans. So we're extrapolating using the conditions we know to exist from different models in different parts of the oceans, and coming up with global plankton biogeographies as an extrapolation. So the oceans, we heard from Minhan that the oceans really are important for everybody. These are they provide us with food. They help us combat climate change. They're possibly impacted by climate change. They provide oxygen, they remove carbon. They provide leisure activities, and they support our transport system. So they're really important for everybody. So the science around oceans really has to be global. There are a couple of important discussions going on at the moment politically from the Convention on Biological Diversity and from other parts of the UN around how we handle scientific data that relate to marine environments. So for the CBD, it relates to coastal environments. For the UN process, this is for the high seas outside those areas of national jurisdiction. And really, it's working out how, in a world where you have wealthy countries and countries that have lower incomes and are less able to engage so easily with science, and certainly scientific exploration, which is a costly endeavor, of course, how everyone can benefit from that system. And I believe very strongly that actually the openness of the data, providing global accessibility to the data from all of these studies, is really an important element of allowing the benefits of this work to be shared with everybody. Part of that is the data, part of that is allowing scientists who are contributing to be credited for what they do and to have attribution. And a part of it is really building a globally connected scientific community to work on these data sets. So finally, I look back to the impact so far that Tara Oceans has had. Here we're looking at some of the higher profile publications, special issues from the project. So from the consortium itself, it's a big consortium, but we had some over 130 publications. And you do go and have a look in here. There's an awful lot of reading material here to get stuck into, but some very interesting science. But what I want to focus on is really a selection of examples of studies that don't come from the consortium. These studies come from other scientists who have picked up the Tara data through the infrastructure that I've presented and developed new scientific ideas and explored more. I won't go into huge detail because I'm running out of time. Here we are looking at, for example, discovery of a new group of rhodopsins. Here we're looking at biomass distribution across the oceans and elsewhere. Here we're looking at selection and in particular how dispersal is restricted by environmental conditions in the plankton. Here we're looking at the diversity of phages across the world's oceans. Here we're looking at the impact of particular codon usage adaptations or codon usage changes in protecting against certain resource-related characteristics of organisms. And here we're looking at using Tara to compare with data from over 100 years ago to look at the calcification, the extent of calcification in foraminifera. So looking at the impact of climate change, or anthropogenic climate change on these calcifying plankton. So they're just a selection of the kinds of science that have happened based on the Tara data set so far. So to acknowledge some of the contributors, I can't cover everybody. Many people work on Tara Oceans. Chris Bowler and Stefan Pizon in particular provided some of the slides and some of the materials I've used. The coordination group, the expeditions group, and I mentioned as well Blue Cloud. It's a broad project, but the demonstrator that I mentioned comes from EBI, Sorbonne University, and FLIS in Belgium. And just on the right to mention some other projects, some of which we mentioned already. Atlantico is ongoing and brings together 5 flagship cruises, and of course we've heard already about the planetary biology program and the ecosystems interest from EMBL. And I thank you for your attention.
All right, great. Thank you for an excellent talk there, Guy. All right, so I believe we're gonna convene now and have our panelist discussion session. Do we have Minhan there?
Yeah.
Okay, great. And before I kick this off, I should also note, as the questions are coming in, that I'm actually a developmental biologist. So I see my role here as really kind of lowering the bar for the discussion. And hopefully a nice way. So don't be scared about putting on questions here. It's a very approachable audience. So I think one of the first things I'd be very interested in hearing, and I think just for the audience who wasn't part of it, this actually started as a discussion among the panelists, is about technology, right? I think a common thread that we saw through many of these talks was really, the explosion of data from Guy's perspective, sampling very broadly. We have Nicole, who's really doing these very beautiful multi-omics studies, really kind of channeling down to these questions, and even the in silico exploration from Minhan. So really what my question is then is really, how do you see this technology really changing kind of the scale and the depth and the scope of the kind of questions that we can actually explore now in these marine environments? So I guess maybe Guy, I would take your perspective first there, starting as in very broadly and then kind of going out through the other panelists here.
Here we go. Yeah. So the thing is, everything is in flux. And although we look today, I think Nicole said that we're in truly a golden age for marine biology. but actually, we may well be in an even more golden age in a few years' time. But what we have now, what we can say is what we have now is gives us so much more power in our analyses and so much more depth than we had 10, 20 years ago. So I think the challenge actually is to make sure that these things don't run away with themselves and we don't lose the ability to track what is happening and to translate the technologies into things that bring those technologies on board the existing programs. So there's a huge, of course, already investment in ongoing monitoring of the oceans, and this is in the life sciences, but also in the physical sciences and the environmental sciences. Making sure that the new technologies are applied and available to all of this, that we really build a global stream of data that we can all tap into.
Great, Nicole, you're Minhan.
Justin, yes, definitely technology driven. For marine research, there's the omics and the imaging, but that's the same, at least in the life sciences, for terrestrial and limnic. What is unique for the marine realm is our ability to go deep and to go broad. So deep is definitely the technology with submersibles, but also with unmanned, autonomous underwater vehicles, but going broad with satellites and some of the wonderful data that we saw from Minhan. This is all the other huge broad side that we're seeing. And then bringing that together with large scale expeditions like the Terra Oceans, where we can go in and specifically also sample data. Or you might have seen in the New York Times, there was an article a few days ago about this huge bioluminescent bloom, I forgot, somewhere in Southeast Asia, just beautiful images. And they last for 45 days. And that's long enough for us to get a research vessel out there and to go and collect it. That is why, maybe it's certainly the golden era. And all the data, though, I agree, will probably take decades, even longer to work up. Yeah, great time.
Yeah, Justin, it's an important question. I think, first of all, it's quite obvious, ocean science, the marine science is data-driven science. Without data, you wouldn't have any ocean science. And the technology and the ocean technology, ocean science is becoming more and more coherent. And having said so, if you look at this, the history of the ocean exploration and ocean research, the most rapid growing data set is from genomic and remote sensing. So that's 1 year of data probably exceeding the old-fashioned ship-based measurement for 100 years. But still, I think if you look at this volume of the ocean data, it's still not yet big data. So it's still a long way to go. And the challenge for the ocean science is that we need to provide solution. And ocean science as now is enough for diagnose the problem, the mechanism, and the process stuff like that. But to solve the real problem, I think we still need the real integration of science and technology, data and the modeling, in order to predict at the fine scale, temporary, and the spatial scale. That's the real challenge. And they say exactly the UN Decade of Ocean Science for Sustainable Development, and try to transform the diagnostic research into the solution-oriented science. I think I would like to add this.
Yeah, okay, fantastic, yeah. So I think another common kind of thread is really, because we're kind of transversing these scales, I think as each of you have really kind of noted. So really, what would the role then of kind of applying the in silico model-based approaches, like how do we really grasp this data, right? Because I think we're struggling this constantly as a developmental biologist. So, and I see the problem even as larger if you consider the global ramifications of what you guys are discussing. So I would just love to hear your comments, and I see you nodding your head there, Nicole. So I'll push it towards you first.
Yeah, I think that's something that every discipline is struggling with. And I think I think as so often, there's never one approach that's going to help us. And I think it's critical, for example, in marine research that we continue to have taxonomists that can identify organisms based on their morphology, because those are the ones that you can go out to in the field without any technology at all and actually find the organisms you're interested in. Well, that's not big data. That's very small data, very small analyses, but very valuable. And of course, there's no question that we all see the value in analyzing the large data, but I think it's always the combination. I think some of the biggest discoveries we've made, whether it's in biology, chemistry, or physics, have always been driven by people looking at something that seems pretty irrelevant, that haven't been driven by trying to analyze large amounts of data, at least in biology. I have to be careful, physics is different, and that are trying to understand a phenomenon. And that's often, how should I say it? So far, a lot of large data is descriptive. You're looking at the data, you're analyzing that, and you're trying to develop hypotheses. But then if you want to test those hypotheses and go down, that's where you're starting to focus down and drive down again. So I think it's a combination.
Great. Guy, do you have any further comments then?
Yeah, just something, I didn't mention this about Tara, but I guess what we're doing perhaps more than before here is we're combining observation with experiment, or with broad observation with specific question. So vessels like Tara they're fortunate in that they are fairly maneuverable. Their programs are not absolutely fixed. They have certain dates and certain ports, but they have some freedom. So they have the opportunity, and Tara has scientists on board who are directing the sampling. So they have an opportunity to, when they see something of interest. So getting out to that bloom within the days that it's there is a fantastic model. And on Tara, it was possible to identify a particular water body of interest and track that body, or stay in one place to monitor. So there are all sorts of things that you can do if you can allow yourself, perhaps in that more traditional experimental way, to modify the question you're asking and modify the sampling routine as you go along. And that's not something you can do in some of the very big autonomous programs, but I think having that opportunity is really important, where you do have this overall observation, but you spot patterns and you go in further.
Great. And I guess, Minhan, this would also reflect in some of your work, right? Where I think we're touching on things where I'd be curious, the scales where you have biology, and then how the biology is actually affecting things like the carbon cycles and all of this, right? So we know these are changing very rapidly right now. So to what degree do you think the biology then is actually driving or impacting a lot of the different cycles you touched on during your talk today?
Yeah, of course, biology is everything. In particular, microbial biology, you see everywhere. You just cannot avoid it. And but we are at very beginning stage to understand the biological function in the global original carbon cycle at just very, very much the beginning. And at the regional base, probably for example, I give one of the example of the Baltic Sea. You see the seasonal pattern essentially driven by this biological activity that drives the cycle of the CO2. But still, if you look at the other region at the global scale, we don't even know the partitioning. If you look at the carbon cycle, the partition biological pump and the physical pump. We just don't know in the quantitative way. And even more than that, it's if you look at the efficiency of the biological pump, that's the export of the carbon from the surface ocean into the deep ocean. you see you are looking at 100% of the error bars. So really, we need a lot more biological observation. I think we were organizing one of these Ocean Ops 19 back almost 2 years now from Hawaii. One is the most talked aspect is biological function and how they place in the carbon cycle. So yes, biology. I'm a chemist as well.
It's a good answer, makes me happy. How about I think kind of a related question to broaden it slightly, and I think Guy touched on this briefly, was that we are talking about these very global issues and problems, and I think many of us sit at rather relatively privileged positions. So how do you think we engage a much broader public to really go after these challenges? I mean, you could do this through either like the data perspective, as Guy was mentioning, to release a lot as broadly, or to be sampling. I would just be interested in a kind of high level what you guys would propose here. Yeah, Nicole, you make the most faces, so you're the easy target first.
Yeah, that's what my students tell me too. Yeah, I'm going to struggle with this. The reason being that the pandemic has made it very clear the struggle scientists face when they're asked to engage with the public. And if I understood your question correctly, you're asking us as scientists. And I work in the Max Planck Institute, and we do basic research, and we're asked to engage with the public, and I feel that's very important, but there are limits to our ability to engage with the public. And I think that we're torn as, this is a very personal opinion, I think we're torn as scientists because we would like to express our deep, deep care for the oceans, their future, their health, and to make it clear why that's important and why we care. But we also are researchers at heart. And so finding that balance is extremely challenging. That's why I'm rolling my eyes, because it's such a struggle to figure out how to do that right.
Yeah, no, I mean, this is in many ways, I think it's probably a very personal question for many of us, right? I think we all struggle with how we can do the best possible science while at the same time, these are serious issues the world's struggling with, people are struggling with. So yeah, I think it's a challenge. So I think it's nice to hear that answer, actually. Yeah.
We all know that problem.
A huge challenge, yeah.
Anybody, is there any other points then? Because I think we're going to go into the Q&A session very quickly here. So if there's any last comments any of you would like to make, then as kind of the panelist perspective, I'd be happy to entertain that.
Maybe just really quickly that I think sometimes it's just enough to just love the oceans, because the oceans are very, anybody who's been to the oceans who experiences them experiences something very deep and very emotional. And I think that's something we can do as scientists also, is work with that, with people that can feel that and say, yeah, and we can actually explain a lot of the biology, the chemistry, and the physics behind why they're so important. But also, it's just wonderful that we have the oceans. It's an emotional statement, but it's an important one for the public.
Yeah, fantastic. At this point, then, I think we're going to open it up more broadly to the public. So again, feel free to keep asking your questions. Going to go to the, let's see, what's the first question here? Okay, this is a good question, and people keep recurring back to this. Recently in the U.S., there was a Hurricane Ida in Louisiana. Do the oceans play an important role in extreme weather? So So I think Minhan, you could probably comment very directly on this one.
The short answer is yes, definitely yes. And one of the reasons because the hurricane, the energy of hurricane comes from the ocean. So when you have the warmer ocean, the hurricane essentially AD will pick up more energy into this hurricane system. That's caused the enhanced strength. So if we look back, there is some statistical study during the last 20 or 30 years. The strength of the hurricane is increased, not the frequency of the hurricane. So that's a strong indication the extreme weather is affected by the ocean. But as a matter of fact, the global scale, the warming is the fundamental reason factor drives all the extreme weather. That's become more and more clearer. If you read the assessment IPCC AR6 just released a few weeks ago, that's a very strong evidence and it become clearer and clearer. So yes.
All right, fantastic. And I actually had the same question, Nicole. Just I think it was a nice one from was it Aubrey Weigel, which says that there are parallels. Are there parallels? Is the question between the interaction between the bacteriocytes and symbions and the human gut microbiome?
Yes and no. There are always parallels. One of the outcomes of analyzing what drives the composition and the strain diversity in the human microbiome and the question asked has been, is it the relatedness of the microorganisms to each other or the environment that they're in, and the importance of that affecting the genes that they exchange with each other. That's called horizontal gene transfer, and that's a huge, huge topic. For example, for antimicrobial resistance, we know that when you take lots of antibiotics, that the microbes in your gut will start to exchange them, other microorganisms will get them, and then what that will do is increase the resistance of your microbial community. We see the exact same thing in our symbioses, which is that it's like there's a whole pool of jumping genes that are around, and they're driven by the environment. So your environment, whether it's in your gut or whether it's a hydrothermal vent, will affect the microbial community. And those are parallels. In terms of many other aspects, of course, they're very different.
All right, great. Minhan, did you have you have your hand up? I wasn't sure if you had a point to make, or is this just a? Okay, that's it. And I guess in a very related question also for you, Nicole. This is from Patishka. Sorry if I get the name incorrect. If these symbionts are able to fix CO2, then can we look forward to them in controlling climate change?
Yeah, actually, I had to think about that. I saw it. I was going, how can I explain that well? Okay. If you want to control global climate, then the CO2 fix, that has to be a net CO2 fixation, has to overall, you have to think about what's the input that came into the environment, and overall is that environment fixing it. The only sites where you would have a net CO2 fixation would be where the energy sources that the microbes are using are geothermically driven. That's critical, and that would be hydrothermal vents or subduction zones. There's simply not enough hydrothermal vents at the bottom of the sea to have enough of these animal communities and to fix CO2. If the energy sources are biologically created, if they're created by microorganisms, then that's actually coming from CO2 fixation through photosynthesis. That's the original source. So you can't So most of dark energy is not going to give you a net CO2 fixation. Also, for the animals, critical, they need oxygen and the microbes too. So if you don't have photosynthesis producing oxygen, these ecosystems in the deep sea are going to die. We estimate if snowball Earth creates a dark Earth, it'll take about, actually Minhan can probably correct me if the estimates are correct, but about 4,000 years until there will no longer be enough oxygen in the deep sea for these communities to thrive. I don't know what the estimates are. The most recent one, that's about 10 years old, where I got that information. So life on Earth is dependent on the sun, and we can't do massive CO2 fixation through chemosynthesis.
All right, great. And the second question. Oh, do you have that? Yeah.
Yeah, can I add? I just want to add Nicole's comments, and I think there is two important lines of the consideration when we talk about the mitigation climate by fixing CO2. In short time scale, because you have to draw down the air CO2. That's the thing plays a role in climate change. So that's the ocean biological pump, physical pump, or enhance the buffer capacity. In the longer time scale, you have to have something change the size of the pore of atmosphere, ocean, and the land. So that takes many years. So it's not a single process. I fully agree with Nicholas, only fixation and the recycling. And one additional point is if you change the recycling rate, for example, the feature of the dissolved organic carbon, in long run, it could be play a role in the climate system by changing the relative size of the atmosphere, ocean, and the land.
Great, thank you very much for the very nice answers here. And this goes for Guy from Khalid Abdullah. Thank you very much. Do you have any data about the Arabian Gulf?
Yeah, I was actually, I meant to click the answer in text. So the answer is yes. It's yes from the Tara projects. It's also yes from other studies as well. And I can help you to find those. So there are ways of searching for those data and bounding a particular region. So do get in touch if you want some support in accessing those data as well.
And I have a question here from, this is actually for Cornelius. I'll throw him under the bus here, but this is, I think, a very good question. And I think one of these things that many of us have actually thought about, which is that if you headed, do you want, oh, go ahead and ask it directly, Cornelius. I think it's a great question.
Justin, which one? I put a couple questions.
No, I like the funding agency.
Which one did you like?
Here. The funding agency is a fantastic question.
Great. So yeah, I was as a way to push the panelists a little bit, you know, in prioritizing research. If you were the head of a national funding agency, in which type of marine research would you put most of your money if your primary aim was to mitigate climate change specifically now?
And they go quiet. You pushed him too far, Cornelius.
Well, maybe I'm going to call on Minhan. I mean, you talked about this directly, so right. Some
the
things you talked about
problem is we are not funding agency. Thank you. It's a tough question, but I think it's a kind of international movement, and it's also true in China. The funding agency is looking for the solution to mitigate or solve the climate change problem in the ocean science. So now in China, we just launched, actually I was the panel committee director, committee chair for this carbon neutrality project. This NSF China just launched. And so they're looking for how China could respond to the carbon emission, carbon CO2 peak in 2030, and a carbon neutrality in 26. So in overall, they try to link this climate change and by providing this strategic solution, policy, stuff like that. So that's the thing they are looking for it. And if we go beyond the climate change, of course, it's a very much they are looking for facilitate, accelerated for marine blue economy, stuff like that. So that's I see the transformative way of doing research, including the ocean science anyway. And the pure curiosity driven, I'm not sure it's good for bad or bad, but pure curiosity is getting more and more difficult to get funded. That's quite, to me, it's a quite clear trend.
So just to be clear, then you think that's a bad trend? As someone who does a lot of curiosity-driven research.
Yeah, but we could get some of the synergy between curiosity-driven and the scientist has to find a way to meet the challenge, to face the challenge the society, the funding agency, or the government is asking for. At the same time, we keep our own curiosity, motivation, and I think there is a way to make it both feasible.
And I guess if I could ask, well, is there further comments before I ask a follow-up for this? I mean, so really this question stems not directly necessarily from what we have in the chat, but a lot of, I was going around asking other people for questions. And I think Minhan, you'd be very good at addressing this. A lot of people really are quite scared after seeing all these reports coming out, right? I think many of the general public who might be involved, how scared should we be as scientists, and what can we do to kind of engage the public maybe? I know we touched on this briefly, so we don't have to comment further really on that. But then, yeah, I think what ray of hope do we have here as a community? I'm looking for some optimism here as we close out the session.
No, I would say there's nothing wrong with that. I think if you look at this, all the motivation, purpose, objective of the research is to innovation, is to try to find something new. So if you look at the ways for innovation, you have this all the discipline driven, that's the curiosity one, and also need-based or solution-oriented or new technology interdisciplinary. I think this is the transformation is not necessarily wrong, and just so we need to be realized and stay on the top of this frontier, and also try to identify the real societal problem. And then the discipline-driven, I would say the original, the traditional curiosity-driven is still there. And the basic research, because there's, in particular for the ocean science, as I said, the ocean is larger and exploited. So there's always innovation potential when you do anything. Just a matter of that you can take care of both sides, need or solution-oriented and the need-based. I don't see it's a negative one.
All right, great answer then. Guy, do you have anything?
I can make a comment slightly to this, but also to the engagement side that we discussed before. Okay, so we've had a terrible time over the last year and a half with COVID. The world has really suffered, but it's sort of pushed some responses from different parts of society. And I spent actually most of my time over the last year and a half has been spent more on COVID data and doing that than on marine data. But what we've seen, I think, is a response by several responses, the scientific community, the number of people who were able to to put aside what they were working on and switch over and understand the problem. And now, of course, a lot of people have done things that didn't particularly lead anywhere, but there have been a lot of contributions and there's been a huge energy. And so the scientific community has been able to engage in a crisis moment with the topic and really learn and develop and work together. We've also seen, I think, a much better engagement from people who are not scientists in understanding data. We now watch on our TV screens, we look at statistics and we see plots far more than we used to, and we're familiar with them. We talk about different metrics and different values. So I think I think we can be a little inspired that the world is actually ready to understand and engage with science, and it can do that. Now, we don't want to wait for a crisis, but arguably actually with climate change, we're sort of in a crisis already. I think it will be perhaps naive to think that we will suddenly turn around and face climate change in the same way we turned around and faced COVID. But surely there are some positives there, that there are points of engagement from the scientific community and from civil society that can be built upon and can be reused and can contribute in some way. Because I do think that the solution to this is it will come from multiple angles. If we don't, we have to get the whole world working together. So I mentioned in my presentation about the access and benefit sharing. It is really important that countries of all economic statuses can participate, because then they can be part of the solution and benefit from the fact that we've solved this problem globally together.
Great, fantastic. And I believe we're actually going over time here. So I think we'll close out the Q&A session at this point. I'd encourage audience members, if you still have any questions, all these panelists are very, very friendly, so you can probably reach out to them directly.
Wonderful. Thanks so much. Thank you, Justin, for moderating a really great session today. As we close out, I just want to remind all of the trainees to please stay on the call for another 15 minutes or so for a special Meet the Panelists session. Minhan, Nicole, Guy, and Justin, thank you so much for being with us today and for some really inspiring talks and discussion. It was really, really interesting session. A big thank you to the audience as always. I hope that you will come back and join us again on October 6th at 9 a.m. Eastern. Our next Life Science Across the Globe is focused on plant biology, and it's being hosted by the University of Buenos Aires and CONICET in Argentina. So we're looking forward to that, and we hope you'll join us. So with that, this concludes the main session for today, and trainees, please, stay on the call. We'll get started in about, I would say, 30 seconds. Give everyone a chance to leave if they want to leave or stay on, take a stretch, and then we'll get started with our Meet the Panelists session. Thanks, everyone. All right, so it looks like we have about 40 trainees